Hello, everyone, and welcome to today's webinar titled SARS-CoV-2, Quality Control for Molecular and Serology Assays. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Laboratories. I'd like to invite everyone to participate by submitting your questions during this presentation. To do so, simply type them into the Ask a Question box located on the far left of your screen and click Send. Any questions we don't answer during the webinar will be captured and responded through the Q&A list that will be posted next week in the Event Resources box, which is located in the Ask a Question box. You'll also be able to download this presentation handouts from this event resource box once the webinar is available on demand. If you have any trouble seeing or hearing this presentation, simply click on the concerns button, Ask a Question box, and we will address those issues. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. So let's get started. Today's webinar on quality control for SARS-CoV-2 testing consists of two presentations and a Q&A session at the end. The first presentation will focus on quality control for molecular testing. I now present today's first speaker, Dr. Duane Newton. Dr. Newton is the director of the Clinical Microbiology Laboratory as well as associate director of the Division of Clinical Pathology, both in the Department of Pathology at Michigan Medicine in Ann Arbor, Michigan, USA. His research interests lie in the development of molecular assays for the diagnosis and management of infectious diseases. For a complete biography of Dr. Newton, please visit the Biography tab at the top of your screen. Dr. Newton, welcome. You may now begin your presentation, sir.
Thank you so much, Susie, and thank you everyone for joining us today. My talk is entitled Molecular QC for Qualitative SARS-CoV-2 Testing. You can observe a lot just by watching. So I'll get into some of these little quotes and their origins here as we get going on the talk. So first off, I'd like to share some disclosures, outside activities I'm involved in. I serve as a consultant part-time with NaviDx. I'm on the scientific advisory board for two companies, Quantimatrix and Biorad, and I'll actually be joining NaviDx as a full-time consultant in September. I'll be leaving the University of Michigan. Couple of objectives today, goals for you in the audience, which I hope you'll be able to take from the presentation, is to be able to describe some approaches for SARS-CoV-2 molecular testing in the quality control standpoint and how that can be used to monitor the entire testing process, as well as look at some optimal practices and maybe ideal characteristics of quality control material that you could use in your assays. Now, first, we have a lot of different directions that we can go for QC, but we might not actually know exactly the direction that we need to go. And fortunately, we have a lot of guidance that can help us. Maybe a little more guidance than Yogi Berra, that baseball great and sometimes trying to be baseball philosopher might say, if you don't know where you're going, you'll end up someplace else. Well, we don't want to end up someplace else. We want to end up in a place where we're performing good quality control, and we're also, we have a robust QC or QA program. And because of the guidance that we have in our labs, in particular from checklists like that's available from the College of American Pathologists, that helps point us down the right road and keep us on the right path. So I'll share with you a couple of items that I'm sure most of us are familiar with, but form the basis for how we perform QC in our laboratories. And you can see in this item, the CAP states that QC should be run at least daily or more frequently, depending on what the manufacturer's package insert says. And that can be impacted, that frequency can be impacted by any changes that occur. And so if there's a potential for change, if there's a potential for variability in your assay, then there's the desire is to do testing more frequently, to do QC testing more frequently. And so any changes of critical reagents, any preventive maintenance that occurs, or components that are critical for performance of the test, any type of change like that, you should do QC again to make sure that everything's operating properly. As far as the types of controls to use, if you're performing a quantitative test, which we're not really going to get into at all today, but CAP gives guidance on QC for quantitative tests, and that should include a negative control, a low positive, and a high positive control. For us, really, in today's discussion, with qualitative tests, a positive and negative control should be run at least daily. What I'll show you some examples of these types of controls, both those that are included within manufacturers' kits, as well as some external controls that potentially could be used for QC and laboratory developed tests. And you'll see that even though they are a positive or negative control, their characteristics can be very different. And we'll talk about some of the reasons why, and maybe some of the attributes that you would look for in an ideal control. But ultimately, the controls that you use must assess the adequacy of your extraction, your amplification, and detection processes. And so it needs to go through the entire testing process to be appropriately used. So as I just mentioned, there are controls that are available from the manufacturer that are included in kits. And there are also controls that are available from third parties that can be used as external control material. But which of these should we be using and when should we be using them? And so sometimes when we look at this, there's lots of different options and potentially forks in the road. And as Mr. Barra says, when you come to a fork in the road, take it. And in this case, we're going to be using both of them or could be using both of them. And so how might that look and what does that look like? One of the challenges, though, especially with SARS controls, is that we need to understand a little bit of the complexity of the virus itself to know how these controls are configured and how they might be used. So I'm going to spend just a brief period discussing the genomic structure of SARS, of the SARS-CoV-2 virus, and talk about the genes and some of the proteins that they encode. And that'll be important for both an understanding of the molecular controls as well as some of the control material that Dr. Theel will talk about in her presentation. So the genomic structure is pretty simple. It's about 30,000 bases. The virus is a single-stranded RNA virus that is an envelope virus. So the first open reading frame contains some proteases that are involved in post-translational modification of the viral protein, so open reading frame 1A. There's also open reading frame 1B that contains the genes for the RNA-dependent RNA polymerase, or NSP12, as well as a helicase enzyme, NSP13. And then there are structural proteins that are involved in creating those protein structures that are involved in the both the envelope as well as with the matrix, the nucleocapsid proteins associated with nucleic acid. So the spike gene encodes the spike protein that forms those characteristic coronavirus crown-like spikes. There's envelope gene for envelope proteins, membrane proteins, and nucleocapsid proteins. So again, about 30,000 bases. The assays and the control material that I'm going to describe are controls that I have used and have been used in our laboratory, and the assays that we have in production in our laboratory, and are meant to be just an example of the kinds of variability and the approaches that manufacturers have taken. It's not an exhaustive list. It doesn't represent all the different options that are available, but are the ones that I can speak to because of our experience. The first assay I'm going to talk about is the Abbott M2000 assay, and this is a platform that we are using for high throughput testing, primarily of asymptomatic patients asymptomatic screening in the pre-procedural arena. The assay targets for this test are it's a dual target, RNA-dependent RNA polymerase, as well as N gene that are the SARS-CoV-2 targets. And then it also includes an internal control sequence that is proprietary, but is a plant pumpkin seed gene that should not be present in any human specimens, but gets spiked into all the specimens that are extracted. It's in the lysis buffer that comes with the system, and it is carried through from extraction through amplification and detection, and it serves to show that the extraction process works, the amplification and detection process works. So it's reported out for every specimen. Within the assay, it has positive control and negative control material. The positive control material is recombinant Synbis virus that contains SARS-CoV-2 sequences that are specifically targeted by the assay. So it includes the RDRP gene as well as the N gene. That virus is replication defective and heat inactivated, so it's not infectious, but it has a structure that's very similar to what a routine virus would appear in a clinical specimen. The negative control is just buffer, that's assay buffer, but that's not really like, I think one of the downsides in this is that it's not like sample matrix. And so I think one of the themes that I'd like to try to articulate through this is the types of controls and the matrices that are used in them should, in an ideal situation, would mimic as closely as possible what you would expect to see in a clinical sample. And that would give you a true sense of how your assay is performing. The other mainstay in our laboratory for SARS-CoV-2 testing is the Diasorin Simplexa test. And this platform we are primarily using for testing of symptomatic patients in our emergency department and inpatient populations. This assay also is a multiplex targeted assay, targeting sequences in the open reading frame 1A and B, as well as the spike gene. It also has an internal control sequence that is proprietary. The nice thing about this assay is that it doesn't require any extraction. You put your sample directly into the disk, and it uses heat to lyse the sample and release the nucleic acid. And then the nucleic acid is amplified and detected. That RNA internal control sequence is part of the master mix that's included in the assay, and so we'll go through that entire testing process as well. The positive controls are a little bit different. These are just synthetic gene fragments of the targets of the assay placed in universal transport media. So it's not like the Abbott assay that's structured like a virus, but just constitutes the target that the assay is trying to detect. And the no template control or negative control that's used in this test is just viral transport media that you have in your laboratory. So that's more like what we would see in a clinical specimen because that's how the samples are getting to us. But the positive control is a little different than what we would expect to see a viral structure taking in a clinical sample. So those are the examples of the assays that we use in our lab, two of the assays that we use in our lab and the controls that are available. I'm going to go through some what I'd like to refer to as external controls or third-party controls that could be used in tests and have a variety of different use cases, which I'll share with you in a few slides. But there are a variety of manufacturers that have developed control material that can be used in SARS-CoV-2 testing. They all have different characteristics. So the two that are listed here are similar. They both originate from cultures of virus. And the one from Zeptometrix is actually heat inactivated fluid from a viral culture. So the virus was cultivated in vitro, and the culture supernatant was taken and heat inactivated, and that's where the high concentrations of virus are present. The virus was actually isolated from a patient in Washington State who returned from China, one of the original patients that were identified in the United States. And that virus has been deposited into repository, and they've provided the specific virus that was derived from. The matrix that it comes in is tissue culture media, which is similar to what a specimen would be received in. Again, this is live virus. Well, the challenge with this is we don't know from lot to lot how much we're getting, because the titer, the amount of virus that's present, is lot specific. The other challenge with this is that the quantity of virus is provided in tissue culture infected doses, 50 TCID 50, as opposed to copies per mil, which we're more familiar with in molecular diagnostics. And so it's difficult to make a correlation between the amount of virus that's present and how much and how that translates into a molecular assay. The BEI product is similar. It was derived from culture, but they actually extracted the RNA. And that is provided in Tris ETA buffer. The biggest challenge with RNA extract is that RNA is very fragile and can be easily degraded by RNases that are in the environment or in other materials that are used in the test. And so there were challenges, we experienced challenges when we used this in freeze thaw and stability over time in dilutions, that the RNA would degrade because it wasn't stabilized. Another product that was available that we evaluated or utilized in our development process was the SeraCare Accuplex positive reference material. This is also, it's very similar and may be exactly what Abbott is using for their positive control, but this is Synbis virus background that's replication defective and heat treated. And within that virus is synthetic genomic information reflected here in this list. So there are different parts of the open reading frame, RDRP genes, the S gene, E gene, and N gene that are designed to be detectable in a variety of assays. In the back, the negative reference material, as they describe it, is Synbis virus with a human RNase P gene. And that is a great negative reference material or negative control, because there are several assays that have been developed that use human RNase P as a target for their internal control to show that you actually had a specimen in the sample, and amplification of that shows that there was clinical material present. And so that's a nice control to mimic what we would see in a clinical sample. Again, these are provided in viral transport media, which are similar to how a sample would be received. The challenge or the limitation with this product is that it's relatively low copy number, and so that makes it difficult to use for limited detection assays or dilution series where you want to have a broader range of concentrations of target. Another product which is available from Exact Diagnostics is synthetic RNA transcripts at a very high concentration, but with large portions of all of the genes listed here. And so they're covering a lot of the genome. And as far as I can tell, all of the assays that are present can be, will have targets that can be detected using this control. The high concentration makes it very easy to get a broad range of dilutions, and the concentration has been accurately quantified using droplet digital PCR. And so the number that you get is very, very precise. It also contains in the background human genomic DNA. And as I mentioned before, there are many assays that target RNase P gene in the human genome as an internal control. And so by including that genomic DNA, you allow for that to be detected as well by the same control material. And it also provides some background that is very similar to what we would expect to see in a clinical specimen, that you'd have target RNA as well as human genomic DNA present in that specimen. So that is more like a true patient sample. The biggest limitation that we saw early on was that the fact that these were RNA transcripts, again, made them susceptible to degradation. And so one of the things that helps prevent that from happening is actually making the dilutions of this in the negative control buffer, because the buffer has RNase inhibiting properties, and so it maintains and improves the stability of that material. And lastly, one of the controls that are widely available and was available very early on was from integrated DNA technologies. And these control materials were simply plasmids containing some of the SARS-CoV-2 genes. And so the plasmids from IDT have the entire nucleocapsid gene. And if you recall from the CDC assay, the original CDC assay targeted the N1 and N2 genes of the nucleocapsid, which are included in that material. It also came in very, very high concentration, that hundreds of thousands of copies per microliter. And so that that material could last a long time. Their negative control was, again, sequences of the RNase P gene, which that human RNase P, which is a target of the CDC assay and some other assays that have been made available. But these, again, these were easy to make, came out at, very early in the response, but really don't resemble what we would expect to see in a true patient specimen. And so it's not an ideal type of control, as we'll get to in a little bit. So I'll talk briefly in the last couple of slides about the value of external controls and their use cases. This can be a little challenging sometimes. We have to put our heads, wrap our heads around some of these thoughts. And Yogi Berra had a tough time doing math and articulated as 90% of this game or this effort that we're trying to make is half mental. But if we can sort it out and maybe go through in stepwise fashion, we can get the other half that's not so mental. What can we use, and how do we use the different materials that are available? There's a lot of different options, from the controls and kits, the other commercial controls. Commercial panels are really would think of more and quantitative panels that have different members that are quantified to a very precise level. And those are really more appropriate for quantitative assays. And so in situations where you're actually looking at trying to get linearity or doing calibrations, that's what I would refer to as a commercial panel. You could use patient samples for a lot of these efforts, culture material, but primarily what we're dealing with is on the left side, the kit controls and other commercial controls. So they have value in a lot of processes, not only from the validation or verification, but also depending on whether it's a FDA or EUA authorized kit. Those kit controls would come with the kit. If you're doing a laboratory developed test, you could potentially utilize these external control materials for that daily QC. So just to summarize the information that was on that last slide, these external control material can be used in verification of FDA approved cleared products, validation of laboratory developed tests. But as I said at the beginning, when you have lot-to-lot changes, you could use your kit controls, or you could use external control material from one of these manufacturers, like in the last few slides. And one of the important things to consider is to use external control materials on a regular basis as an independent monitor, to monitor across the lots, to monitor across changes in the instruments or preventive maintenance. And so, when our our kit lots change, we might compare one lot to the other, but how does that compare to lots over time? And if you have a very stable external control material that has a very long out date that you potentially could run across lots for months, that might give you a better indication that there are changes happening to your testing that you might not pick up by just doing lot-to-lot comparisons. These commercially available products are often much more stable than some of the other options that we might make ourselves, like taking patient samples and pulling them and making aliquots and freezing them, which is a lot of work, much more work when we have to create them our own. And if designed ideally, they can really, these external controls can do just as good of a job as mimicking real clinical samples as ones that we would make ourselves. And so if think about, with all this information, what would be the ideal control, I think I've listed a few characteristics. I'd want them to be as close to being a real virus or mimicking a real virus or pathogen as possible, so that it structurally resembles the pathogen that you're trying to detect, and that it has the target information that is broad enough that it could potentially be used with any manufacturer's assays. One of the problems with the SERCare assay, I listed all the gene targets that was in there. It couldn't actually be used with the diasorin test because the targets in the open reading frame that it has in its in the synthetic virus do not overlap the sequences that are targeted by the diasorin test. So that was one of the limitations for there. And so the control material needs to be robust enough to have to be applicable on all these different platforms. I'd like to see high concentrations or high titers that are precisely quantified so that you can do large dilution studies and look at limits of detection and be able to compare across platforms that might have different LODs. We want them to be stable to freeze thaw, because a lot of times when we're doing these tests, you can't do them all in the same day. And so you'd like to make your dilutions and freeze them so that they're it's from the same dilution series. I'd like to see genomic DNA background in more of these controls. That helps with serving as a control for some of the assays that target human genes, but also because that is how our patient samples should ideally be. They should have human DNA in them, and that can impact how our assays perform. I'd like them to have a matrix which mimics the real specimens that we're receiving. And as we stated at the beginning, it controls for the total testing process from extraction to detection. So with that, I'll finish my part of the presentation, but the game's not over, because the very talented Dr. Theel will be taking over and sharing her perspectives on QC for serology testing.
Thank you, Dr. Newton, for that informative presentation on molecular QC for qualitative SARS-CoV-2 testing. Turning our focus now to quality control for serology testing, I now present today's second speaker, Dr. Ellie Theel. And Dr. Theel is a director of the Infectious Diseases Serology Laboratory at Mayo Clinic and an associate professor of laboratory medicine and pathology at Mayo Clinic College of Medicine in Rochester, Minnesota, USA. Dr. Theel oversees the development and evaluation of serologic methods at Mayo Clinic and has evaluated over 15 different serologic tests for detection of antibodies to SARS-CoV-2 over the last few months. For a complete biography of Dr. Theel, please visit the Biography tab at the top of the screen. Dr. Theel, welcome. You may now begin your presentation.
Great. Well, thank you, Dr. Newton, for the informative talk on molecular testing for SARS-CoV-2. And Thank you, Susie, for the introduction. So for the second part of this webinar, I'll talk about quality control considerations for high throughput qualitative serologic tests for SARS-CoV-2 that currently have emergency use authorization. So before delving in, these are my disclosures. And for the learning objectives, they're listed here, and hopefully by the end, similar to what Dr. Newton mentioned, you'll be able to take away a couple of kind of optimal quality control practices for serologic testing in your lab. So when it comes to SARS-CoV serologic assays, 39 currently have EUA, of which about 20 or so are what I would consider high throughput serologic assays. And important to keep in mind is that these are all quite varied in their design, including their format. So we have rapid lateral flow assays, batch testing, enzyme-linked immunosorbent assays or ELISAs, and then your more continuous load chemiluminescent-based assays. The specimen types that have been validated also differ. Most are validated for serum or plasma, although there is now one site-specific lab that can use dried blood spot extracts. The type of antibody that is detected also varies between these assays, be it IgM or IgG or total antibodies without differentiation. The SARS-CoV-2 protein or antigen that's used to capture these antibodies also varies. Some assays use recombinant subunit 1 or subunit 2 antigens or recombinant receptor binding domain regions from the spike protein, and then other assays use a recombinant form of the nucleocapsid protein. And then finally, while the vast majority of EUA assays are qualitative in nature, there is now one semi-quantitative assay that has received EUA. So overall, though, the point here is that these variables should be kept in mind when considering what type of control you will be using for your assay and for tests specifically that don't have control materials provided in the kit itself. So this is just a high-level table summarizing all of those variables that I just mentioned for the high throughput assays in particular. And you can see that the majority of them at this point are actually chemiluminescent based, which unlike for the ELISA-based methods, most of these typically do not come with their own internal kit control. Instead, for these assays, the laboratory either has to create or purchase and validate their controls separately. And so while most of the manufacturers of these assays do provide external QC material for purchase separately, there are also now at least 3 third-party vendors that have also developed quality control materials. And I've just summarized some of their key characteristics of those products here. So for all 3 major vendors, the source is a human-sourced plasma or serum that's in a liquid, ready-to-use format, so you don't need to rehydrate, which is nice. The shelf life for these products is generally pretty long, at least 1 year if unopened, and then ranging anywhere from 10 to 30 days after opening. And I should mention that Bio-Rad is working on extending that open expiration date out to about 60 days. And this open expiration date is important from a workflow perspective because unlike for molecular tests where controls are taken out and typically thawed right before the run is started, for many of these high throughput assays, the controls are loaded and kind of left on the instrument until they're used up or they expire. So you can also see what antibody isotypes these controls are designed to detect. With currently, none of them actually have an IgM specific control reagent that's available. And then I've also included what type of assay these vendors are using to QC their controls before they're released. So you can see that Technopath and Bio-Rad QC their control material using both a nucleocapsid and a spike-based antibody assay, whereas Serocare, at least from what I can tell, is only using a nucleocapsid-based assay to QC their material. So although one would expect or envision that both anti-spike and anti-nucleocapsid antibodies are present, that may not always be the case. And so this is just something to keep in mind if things look kind of wonky, if you will, during the verification of this control for a spike-based assay. And then also, while a target range for these controls is not indicated or really defined, Seracare does state that they intend their positive control to be a low positive for these assays, although you can imagine that this is probably going to vary from assay to assay. So for tests without an in-kit control, there are a number of general factors to consider when selecting the control material. And one of the most important considerations is the type of control material you're going to use. So ideally, that control should be different than the calibrator that's used in the assay. But if there are no other options available, the lab could validate control material, but it would have to be pulled from a different kit lot than the one that's currently in use. Thankfully, for SARS-CoV-2, there are a number of options available for alternative control material. So as I mentioned earlier, assay vendors do offer their own controls, which is nice, but these controls are often really optimized for the assay, and they may be similar in formulation to their calibrator. So as a result, use of these may not as readily detect potential performance issues. So there's these third-party vended controls, on the other hand, which are not typically designed with any specific assay in mind. So they're independent. They're an independent check of assay performance characteristics and are often preferred from a quality assurance, quality management perspective. And then finally, another approach is for labs to validate and use pooled patient samples for control material, which, while less expensive, as Dr. Newton mentioned, there's a number of challenges here. They're often related to stability issues, having sufficient specimen volume depending on the prevalence of the analyte. There could be issues with result consistency, and so on. So other general considerations to think about when choosing a control include making sure that the material you choose matches the assay you're using. It's probably an obvious point, but it's an important one, as this has been overlooked a couple of times in my experience. You also want to strive to use control material in a matrix as close to possible to the patient samples that you're testing. Ideally, that material would be available in large batch sizes to minimize the variability and allow for trending over long periods of time over different kit lots. And so that goes along with the preference that these materials have a relatively long shelf life, and then obviously cost is something that most labs will consider as well. So once a control material is selected, a few common questions typically come up, including how frequently should QC be run. So per the CAP Immunology checklist for non-wave tests, controls should be run at least daily, which is also what most vendors recommend. But they can obviously be run more frequently, and that's typically a call that's made by the laboratory director themselves. So the frequency of control testing is to an extent dependent on the method that's used. So for batch testing, where there is a defined start and stop time during which all the calibrator, control, and patient measurements occur, QC is run with each batch. And so that's typically the case for your ELISA-based assays. On the other hand, for methods that allow for continuous load or continuous testing, there's really not a similar kind of defined time interval during which all these measurements occur. Testing just goes on until reagents need to be replenished, or there's instrument maintenance, or the instrument is shut down for whatever reason. And again, most manufacturers still recommend QC testing just every 24 hours. So there's a number of potential issues with this fairly infrequent control schedule, including the need to do quite an extensive look back if that control fails. And so for that reason, many labs run controls much more frequently. So for example, rerun them about every 8 hours or so. And this type of QC is often referred to as bracketed QC, because if the QC passes, one can assume that the patient measurements that were made between the two QC events or brackets are likely acceptable. And then in general, QC should be run whenever there's any sort of scheduled events that could change the performance of the assay, including things like calibrations or maintenance, or new lot or new calibrator lots. And in those situations, it's actually recommended that QC be performed both before the change to ensure that samples tested up until that point are valid, and then also after the event to confirm that that change did not impact or alter assay performance. So another common question that comes up is whether the numerical or quantitative value of the control matters for qualitative serologic tests in particular, especially since we're just reporting positive or negative results. So as a quick refresher, the current high throughput serologic assays are designed such that qualitative results are determined based on an established signal to cutoff or index value threshold. And so anything above or below that threshold is resulted as positive or negative respectively. And so for such tests, CLSI recommends that at least 2 control levels be used, one above and one below that threshold. And then ideally that positive control level should be near that qualitative cutoff or qualitative threshold, since at that level, the control is much more likely to detect an out-of-control situation compared to a positive control with a much higher relative level of that analyte. So choosing what that control level is or can be tricky, though, because you don't want it to be too close or at that cutoff threshold, since that might lead to control failures and unnecessary rejection of runs. So to overcome this, we typically consider the assay precision characteristics that we've established during the verification and then choose a control level that's about 2 to 3-fold above that cutoff threshold. And then I also just wanted to mention that along these lines, the CAP Immunology checklist has a requirement specifically stating that that qualitative cutoff threshold needs to be verified initially after implementation, and then every 6 months thereafter. And depending on the assay, while some have calibrators that are either near or used to determine that threshold, another way to satisfy this requirement is to use controls that are near that cutoff. So when it comes to establishing the control material target value and range, there's a couple of things to keep in mind. First, while some vendors may provide already pre-assigned values and ranges for their controls, those should really be used as more of a guide and not a replacement for individual laboratory established control means and ranges. And then there are a couple of ways to determine the means and ranges associated with these controls, depending on whether or not the laboratory has historical data available for those particular QC materials. So, for example, since the imprecision of an assay is an innate characteristic of the test, regardless of the quality control material, if there is historic QC data available for that control and that control has performed well without issues in the past, the established SD or standard deviation can be applied to the new QC material if that target level is similar between the 2 lots. So the mean of the new QC lot can be determined using 10 measurements, ideally on separate days, and then you can apply that historic SD value around the new mean. In most cases, though, especially for SARS-CoV-2 controls, there's no historic QC data available because these products are just so new. So in that case, new control means and SD values have to be established prior to implementation of the control material. And as a standard, this is usually established by measuring 10 points. Ideally, those are acquired on separate days to account for as much variability as possible. And then these 20 points can be plotted on a Levy-Jennings chart to look for any drifts or shifts or outliers. And if none are observed, all of those points can be used to establish the mean and SD of that control material. And so this is just an example of what our 20 points look like for the Seracare positive control material that was run on the orthoclinical IgG chemiluminescent assay. And then oftentimes, because of the potential limited reliability of the initially established SD values, we'll often reevaluate the mean and SD about 1 to 2 months into running the controls. This really allows us to take into account additional procedural variations and factors like use of different calibrators, different reagent lots, any preventative maintenance, and so on. So we have a much larger data set with which to work with that would lead to a more realistic and a more accurate SD. And then finally, you'll obviously want to monitor your QC values over time, often again using Levy-Jennings charts and applying the various Westgard rules, including ideally use of both single event QC rules, which will detect kind of large shifts in the assay, or I should say, using counting QC rules, which will detect smaller shifts or trends in the assay over time. So for example, in our lab, we routinely look at 1,3S, 2,2S, and 4,1S failures or trends for our tests. And so that leads me into a very brief kind of case study that involved a control for one of our SARS-CoV-2 serologic tests, again, kind of underscoring the importance of monitoring QC over time. So we implemented a third-party QC material for our Roche total antibody SARS-CoV-2 assay. And after about a month, we noticed that the signal to cutoff ratio for that positive control material was trending downward on all of our cobas E801 instruments. And you can see kind of a bird's eye view of that in these charts. So we performed an investigation to see what was going on and found that for the most part, the negative control signal to cutoff levels remained stable. We also retested previously positive patient samples and found that those signal to cutoff values were stable and within the expected imprecision of the assay. And then also prior to implementation of the vended control material, we were using pooled positive and negative patient controls. And so we pulled those out of the freezer and tested those, and similarly found that the signal to cutoff ratios remained within the expected range. So our conclusion was ultimately that we were probably seeing some sort of degradation of our positive control material, despite it still being within stability. And I will note that this was not the Bio-Rad control. But we ultimately moved to using an alternative vended third-party control. But again, this is just one example of why you'd want to monitor the numerical values of your controls, even if the results you're reporting are simply qualitative, because as you can imagine, over time, this would lead to failed runs and lots of unnecessary lookbacks and investigation. So to conclude, just some general suggestions for antibody-related quality control. Third-party vendor control material is often preferred, similar to the situation for other assays, in order to kind of independently monitor the performance of your tests. For qualitative assays, which are based off of a cutoff threshold, you again need 2 levels of control, with the positive one ideally being near that qualitative threshold, and you'll want to establish and verify your own control ranges. The frequency of control testing will be dependent on the method you use, as well as the laboratory workflow, but it's typically more frequent than the just once per 24 hours. And then finally, just remember the importance of monitoring your QC for trending over time in order to avoid potentially unnecessary assay control failures. And so with that, I will thank you for your attention and turn it back to Susie.
And thank you, Dr. Theel, for that great presentation. We will now start the Q&A portion of the webinar. If you have any questions you want to ask to either Dr. Newton or Dr. Theel, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. Our first question is for you, Dr. Newton. What is the purpose of including genomic human DNA in SARS-CoV-2 control?
Thanks, Susie. It's a really good question. And there are a variety of assays that are available, including the original CDC SARS-CoV-2 assay that use human targets as internal controls for their assay. So those target genes, like the RNase P gene, are present in that genomic material. And so those would be present in a clinical specimen. And so for our external controls or any kit controls that have genomic material in it, they would be able to detect those targets as well. And I think it also, as alluded to in my description of an ideal control material, because of that genomic material would be present in a clinical specimen, having it part of a control would really help to ensure that that control mimics what a real patient specimen would look like.
Our next question is for both of you. As long as my positive QC is positive and negative is negative, why would I worry about shifts or trends for a qualitative test? We'll start with you, Dr. Theel.
Right, so it's a great question because at face value, right, we report positive or negative, and if our controls are positive or negative correctly, then everything should be fine, right? Unfortunately, just a qual pos negative doesn't really tell you much about the analytical performance of your assay. So as in the example case I showed earlier, I think really shows that. So technically the controls we had qualitatively were in. They passed, but clearly something was going on. And if we didn't monitor those quantitative values, I think at some point we would have reached a point where our positive control would have repeatedly tested negative, and we'd have to do quite a bit of kind of backtracking and investigation and failing runs unnecessarily. So for that reason, I think even for qualitative tests, it's helpful to monitor those numeric values, and even if they're not necessarily clinically significant.
Thank you so much, Dr. Theel. And how about you, Dr. Newton?
I agree with what Dr. Theel said, and would just add to it that, in her case description, she described degradation of the product as as the reason why that those trends went down. There are a lot of situations that could happen that would impact the performance of that material. And it really highlights not only the value of looking at the numbers, but to actually have that external control as part of your assay that's carried out over many, many lots, over many, many days, and even potentially across many different instruments, that you'd have something that you can compare to, not just in a positive or negative way, but in a purely quantitative way, to evaluate when those issues or to identify when those issues are coming up. And so those numbers, as Dr. Theel said, are really important, because without that, you don't see that information, you don't see that change as dramatically and as obviously.
Thank you so much. Our next question is for you, Dr. Theel. Is there any guidance available suggesting which targets are preferred for antibody testing? I mean, in terms of nucleocapsid S1, S2, or receptor binding domains? And if not, do you have any recommendations?
Yeah, so that's a good question. So there's many studies now coming out, right, looking at our immune response to these various SARS-CoV-2 proteins and antigens. But kind of at a high level, I think there's a few take-home points that we can take from those. So overall, I don't actually think there's a really meaningful difference in sensitivity, especially during the early stages of infection, between using serologic assays that are based on the nucleocapsid protein versus any of the spike-based protein assays. Regardless of the test, all individuals seroconvert, or not all, most seroconvert 12 to 14 days after symptom onset. From a longevity perspective, I think we're still really learning about how long these antibodies last, especially to the various proteins. There's some recent studies suggesting that maybe antibodies to the nucleocapsid decline faster than those to the spike proteins. But honestly, I think the jury is still out on that. And then even though it's kind of hard to say exactly what will happen in the future and what will be needed once we have vaccines available, many of the vaccines are, targeting or trying to stimulate an immune response to the spike protein in particular. So it may be beneficial to have spike-based assays on board. But again, how those will be used in the future is hard to say.
Thank you so much, Dr. Theel. Our next question is for you, Dr. Newton. What's the best material to evaluate the limit of detection for a method?
Yeah, I think the important thing to recognize first is that different assays ultimately will have different limits of detection. So if you have multiple platforms in your lab that you're evaluating, you need to have a material that is present in a high concentration, a high enough concentration where you can make a wide range of dilutions that allow you to really differentiate those LODs from assay to assay. And as I mentioned in the talk, some of the controls that are available from commercial manufacturers don't come in a high enough concentration to allow you to make broad dilution series, especially. And when you make those dilutions, you're going to spike them into transport media, 1 mil or 3 mils of transport media, which further dilutes the sample. And so the concentration of that dilution needs to be very high when you're spiking it. And so that, I think, is the first thing. The second thing that's really important is that the concentration of the material is really accurate. And so there are a variety of options that are out there that have used droplet digital PCR to quantify that material, and those numbers are very, very accurate and very, very precise. So I think those are the two most important factors that allow you to have high concentrations that are accurately represented, and then finally, that it's a material that can be stable over time, that you can make dilutions, store them, pull them out, and test them again, and get the same results, because you're often doing these experiments on multiple days with multiple individuals and multiple instruments, and you'd like to have the solutions that you're using to make that determination be the same in all of those evaluations. You're not going back and repeating and adding another variable to that process.
Thank you so much, Dr. Newton. And I want to thank our audience for these great questions coming in. We have time for just one more question, and it goes to you, Dr. Theel. Some third-party QC vendors provide interlaboratory comparison reports for their QC. Are there any benefits from participating in such a program for SARS-CoV-2 antibody testing?
Sure. So there are definitely some benefits to participating in programs like that, because it does in some cases provide kind of a real-time intra-laboratory comparison or an evaluation of how your controls are performing and ultimately how your assay is performing. So these sorts of programs basically let the lab see how their assay is performing relative to other laboratories using the same assays and controls. And if you find that your controls are kind of wonky compared to other assays, you'll be able to see that issue fairly quickly and take appropriate measures to investigate the issue before it becomes a major problem. So,
thank you so much. Dr. Newton, Dr. Theel, thank you both for your time today and for your important work and your research. Before we go, I do want to thank the audience again for joining us today and for their interesting questions. And questions we did not have time to address today will be answered and posted in that event resources by the end of next week. This webcast will be available on demand in the auditorium from Friday on. And if you found this information informative and helpful, please share it with your colleagues. Bio-Rad will be hosting a new QC-related webinar every month this year. Next month's topic is how to limit patient harm from erroneous results, QC strategies based on risk management, and will be held in September, on September 16th. Please check back regularly for new topics and take time to explore our posters, booths, and Bio-Rad's new COVID product showcase in the virtual conference. Until next time, take care, be safe, and bye-bye.